Hello everyone, today we're going to have a look at cell wall structure. So, the three things that we're going to be looking at in this lesson is being able to describe the structure of plant cell walls. We're going to be able to explain the role of cellulose, hemicellulose and pectin in the structure and function of cell walls, and we're also going to explain the function of plasmodesmata and pits. When we think about plant cell walls, normally we imagine it as a single layer outside of the plasma membrane that is going to surround the plant cell and grow and help support their structure by being quite strong. We also know at the moment that it's made of a mixture of polysaccharides that are cross-linked together, which can increase properties like flexibility, strength, turgor and waterproofing. So you can imagine this as a structure that looks a bit like scaffolding with lots of different components together that is going to help support that plant and keep it standing up nice and tall. So when we look at individual cells jumping together, what we notice is that we don't just have a single line around the outside. You can see that there's lots and lots of layers around the outside of those plant cells on the left hand side, but also when you've got the properly outside each cell, where it's no longer connected directly, you have this sort of glue holding these three different cells together. And if we look at it in a slightly different way, this time if we're looking at a more tubular shaped vessel on the right hand side, you can see much more clearly that a cross section showing you that there is more than one layer to your cell wall. So let's look through what these different layers are. Firstly, on the inside we have our plasma membrane, and that is just that yellow barrier right here that shows us the outside of the cytoplasm where you have your plasma membrane holding everything together. Outside of that we have this thicker purple layer, and this is called the secondary cell wall. Outside the secondary cell wall we have this green layer, which is called the primary cell wall. And then outside your plant cells, but gluing the different ones together, we have this little yellow substance that looks like the cement between bricks, and that is called the middle lamella. We can see these layers again if we look over on our cross section, where we have number 2 here representing different parts of the secondary wall, so that's a bit more detail than we need really, but it's just to show you that there is a lot of it and it's quite thick. Outside that we have the primary wall, and outside that we have the middle lamella. So here it doesn't look yellow, it looks like a darker colour, and you can imagine lots and lots of these tubes connected together by that middle lamella acting as cement. And let's look at some definitions for these. So starting off with the cell membrane, it's a phospholipid bilayer containing integral and peripheral proteins, and these allow the cell to control the entry and exit of substances. The secondary cell wall is a layer that's stronger and it's for mechanical support, sometimes containing lignin that you might have heard before in xylem. The primary cell wall is the first cell wall layer that's made by a new plant cell, and it starts off soft and bendy. If you imagine a little plant that's just grown from its seed, it will be very, very soft, and then as it grows taller and stronger, that primary cell wall can harden and toughen up as the cell matures. And lastly, we have the middle lamella that we've mentioned already, and this is a layer that holds the cell walls of adjacent plant cells together, a bit like cement. So remembering the pictures that I've showed you and these definitions, what I would like you to do is draw and label a group of adjacent plant cells next door to each other to show all the different layers that we have on the outside of a plant cell. There are 4 things that you should have. Pause the video, give it a go. Right, so let's have a look at my picture. So let's draw our 3 plant cells side by side. We want to show that glue together, and you might want to have a quick check of your diagram to make sure we're matching the same number of layers. Let's start right from the inside. So we have our cell membrane. Just outside the cell membrane we have this layer, which is the secondary cell wall, and outside that we have the primary cell wall, and then gluing all these different cells together we have the middle lamella. What we need to know next is what important molecules are involved in making your cell wall and sticking all of these different layers together. So there's 3 main molecules that we need to pick up on. The first one is a familiar friend. We've heard of cellulose before right at the beginning of our course, but now we're thinking about them bundled together to form something called microfibrils. Then we need to know about something called pectin. And lastly, surrounding the cellulose, we have something called hemicellulose. So we're going to have a look at each in turn, and we're going to start with cellulose microfibrils first. So let's warm our brains up and think about these structures. You can see there's a couple of pictures to show how they would be lying on top of each other in order to make that nice mesh. But first, I'd like you to draw back through your memory to the beginning of the year to draw a molecule of beta glucose. After you've drawn one molecule of beta glucose, I would like you to join many of them together, let's say about 3, to show the bonding between different beta glucose molecules to form a polymer called cellulose. So again, pause the video, grab a bit of scrap paper and give it a draw. Let's see our molecule of beta glucose first. And there's 2 main ways that you could draw this. You could draw the simplified structure on the left hand side, which cuts out a lot of the extra carbons and hydrogens and hydroxide groups that are in the central body here, but keeps the groups that are on the end. Or you could have a bit more of a detailed version on this side. But what we're looking for in beta glucose to make it different from alpha glucose is to look at the position of these hydroxyl groups on either side. They should be opposite in beta glucose compared to alpha, where instead you would have an OH group here and this up top would just be a hydrogen. So if we're bonding them together, we can put these 2 side by side, and what's important is that this molecule, the one after the first, is flipped. It's upside down. So we have a look at where we would make our lovely glycosidic bond here. So you would take a molecule of water out in a condensation reaction, and we would end up joining those 2 together. So let's have a look at how that would look properly. What we can see here in our cellulose structure, again, I've put little boxes around where you have that flipped part, so you could also track the oxygens there to make sure you can see it's alternating, so you can see each beta glucose molecule is flipped, held in place by this little glycosidic bond in the middle, and with each glycosidic bond you have made, you're producing a molecule of water. Something that we haven't mentioned yet is what kind of glycosidic bonds are holding these together. And we need to remember that when we have a glycosidic bond and it's part of the straight chain, that this is called a 1:4 glycosidic bond. I'm just going to write GB here. If we were doing a branch point, so if we were joining a polysaccharide chain, let's say to this position here, and we're making a different branch of glucose molecules, that would make it a 1:6 glycosidic bond. So let's think of how we can describe these cellulose microfibrils we've made. So the microfibril we can see is made of more than one little molecule of cellulose. Before I go through and add more details to it, pause the video here and see how you might describe it. What different structural features do we have? Let's go through. So, pardon me, first off on the last slide, we've got a beta glucose polymer, or a polysaccharide made from beta glucose, joined together by 1:4 glycosidic bonds. We notice they're straight chains, so we can describe that as unbranched, and they're all running parallel to each other, so we can describe the change as parallel. And you notice that there are these little blue lines showing connections between them, and this is representing a kind of bond called a hydrogen bond that is going to hold them together, and having so many of them is really going to help improve its strength. Finally, we can say that we have formed microfibrils. So here's another picture to show that we've got our different monomers, our monosaccharides of beta glucose, bonded together by 1:4 carbon bonds or glycosidic bonds, making a single molecule of cellulose, and then we've got those hydrogen bonds connecting each parallel chain of cellulose in order to form the cellulose microfibril, which is made from many. What we need to look at next is why these structures are important. Why bother with all of these different or very particular structures? So first off, we can focus on the fact that we have formed parallel microfibrils. Why should they be parallel? And the main function of this is partly due to the fact it is found in a plant cell wall, and cell walls we know are very important for structural support. Having those hydrogen bonds allows the cellulose to bond to nearby molecules to form those microfibrils. So if you imagine we cut all of those different bonds together, those strands wouldn't be held together nearly as well, and they'd all fall apart. So if they're falling apart without these hydrogen bonds in place, then it is much weaker as a structure. So having these hydrogen bonds in the cellulose microfibrils keeps it together nice and tight. Next, we are looking at the fact that they're laid down at angles, and this is going to allow cellulose to be strong but flexible. If you imagine the grain of wood, if everything is running one direction and you cut it in the middle, then you'll make a nice break point. But where your cellulose is laid down, let's say you have some going in this direction and some going in the top over it, here now it has a bit more flexibility to be bent, but still retain its structure. There are two more things we can look at. Let's start with pectins. Pectins are a network of polysaccharides found in the cell wall and the middle lamella, and you can see them on the diagram on the left as this network of blue strings wrapping around the cellulose in this purple molecule called hemicellulose. Now hemicellulose by comparison you can see is a lot shorter. There are lots of short branched polysaccharides, and you can see them stuck along the cellulose microfibrils holding different strands together. So if we have a look around, pectin goes all the way through, kind of like a glue cementing anything together, but your hemicellulose is very particular to your cellulose microfibrils connecting different ones together. So this is called cross-linking. And you can imagine it as something that is going to help reinforce your cell wall, the same way that if you have reinforced concrete, you would put that cross-linked network of steel inside in order to, and then reinforce it with concrete in order to make the overall structure stronger. So by having pectins, hemicellulose and the cellulose microfibrils all connected together, you end up with a very strong, steady structure. Now what we're going to look at next is something that you'll have to pause and think about. I've given you a blank diagram in your booklets, and you can notice that there are 3 different layers that we need to name on the left hand side. And on the right hand side we have 1 arrow to these red strings, we have 1 arrow to those larger green rods, and we have another arrow to these thinner and thicker blue filaments running through. So depending on where they're found, you should be able to label up what structure is present where. Let's read out our hints first. So our first clue is that cellulose microfibrils are laid down at angles to one another. The next clue is that there is less hemicellulose in the middle lamella. The next clue is that cellulose microfibrils are held together in a matrix with glue made of hemicellulose and pectins. The next one is pectins are also an important component of the middle lamella, and that the middle lamella is the region between cell walls of adjacent cells. So based on their position, try and find out where you can put your labels for plasma membrane, cellulose microfibril, hemicellulose, pectin or calcium pectate, primary cell wall, and the middle lamella. Let's look at our diagram. So let's go through the different regions first. At the bottom we have our plasma membrane, and then we're looking at just the structure of the primary cell wall, and outside of that we have the middle lamella. Next we notice these green rods that are very large and indeed put down at angles to one another, so these are the cellulose microfibrils. Next we're looking at this red layer, which is very tightly joined to different cellulose microfibrils running all the way through the primary cell wall and just a little bit in the middle lamella, so this is called hemicellulose. And lastly we have our label pectin, because we can see that pectin is a main component of the middle lamella. It's a lot thicker and there is much less hemicellulose if any there, but it's also found in the primary cell wall layer. So now we have our diagram, and we have a few sources of information to read and summarize, and we want to take away 3 points from each. So what I'd like you to do is pause the video when we look at each source and then read it through, pick out the 3 main points and add it to your table. Let's look at the first one. So the main points from your summary here should be that they're created in the first meiotic division, they're the first cell wall to be made, they are thin and they're flexible when that cell is growing, and then at the end, when the cells finish growing, they become stronger as the cell matures. Let's look at our next source. Okay, so the main points from this slide are that they are thicker and stronger than a primary cell wall, they are cross-linked with lignin and hemicellulose, and the secondary cell wall provides a network that allows the cells to be rigid and have some of waterproofing properties from that lignin. Lastly, our middle lamella. In your summary, you should have that they are mainly composed of pectin, that they join adjacent cells together by their cell walls, and that they improve stability and allow plasmodesmata to run through them. So we have our summary, we have some notes that we can work with in order to put some answers together later. But as we've just mentioned plasmodesmata, I thought it would be useful to do a little recap. So plasmodesmata we can see as these tubes connecting two cells together here. So plasmodesmata connect the cytoplasm of cells next door to each other, adjacent cells, to allow substances to move between them. If we look at pits in comparison, we notice that the pit seems to be a lot larger, and you can have plasmodesmata running through it sometimes, but they're just areas of the cell wall that is thinner and allow some transport of substances across and between cells. Now, if you're feeling confident, I'd like you to use your table. Otherwise, I'd like you to hide all your notes and try and answer these questions. We start off nice and easy. What substance is the middle lamella mainly made of? After this, we need to think of an application. What would happen if the middle lamella was digested between different plant cells? And finally, we need to think a bit harder. What are the similarities and differences between a primary and a secondary cell wall? Take your time. You can put it on for 7 minutes, and then we'll go through our answers. Okay, let's look at our first answer. So the middle lamella is mainly made out of pectin. And if the middle lamella was digested, then those cells wouldn't stick to each other. Imagine having a wall made out of bricks without any cement. They would still be there, but they wouldn't be able to be held together very well, so the structural stability would decrease. We need a bit more room for the next one, thinking of the similarities and differences between the primary and secondary cell wall. Now, if you haven't got much of this, you could have a think about what materials would be involved. What is their functions? Is there anything that's different in their functions? Does a primary and a secondary cell wall have the same materials making it up? Is there a difference? If you've not got much, pause again and give it a go. So let's look through our answers. In a primary and a secondary cell wall, we will find cellulose microfibrils, hemicellulose and pectin. All three of those things should be in each one. And as they're both types of cell wall, it makes sense that both of them should provide mechanical support to the plant cell. However, we have two differences that we can point out. A primary cell wall is made first, remember? So this is when your cell is much smaller and it needs to be able to grow and elongate. So if you have a little cell like this at the beginning, it needs to have a nice primary cell wall which is flexible and soft enough to allow that cell to grow and elongate. This isn't true so much for secondary cell walls. Secondary cell walls are much thicker and much stronger, so these are going to contain lignin to add that structural support and strength that a primary cell wall does not have. Score yourself up, and then we want to really test ourselves with these past exam paper questions. So take your time, we'll look through each one. Here we have a picture showing part of a cellulose cell wall through an electron microscope. Using the information in the photograph and your own knowledge, what you need to do is describe the structure of a cellulose cell wall. Let's say this is a 3-mark question. Think of what you can remember about the structure of this cellulose cell wall. Let's look through our potential points. Firstly, we need to say what it's made out of. So, a cellulose cell wall is a polymer of beta-glucose. We have lots of cellulose. Each cellulose molecule is going to be held together by hydrogen bonds, or you could say as microfibrils, and it says ignore lignin quite kindly. Next up, we need to talk about the way that these are laid down. So each microfibril is parallel, but you could also say that layers of microfibrils are arranged in a mesh, or a crisscross. And finally, we could mention the things that hold them together. So there is a matrix, or those cross-links, between hemicellulose and pectin. Score yourself out of 3. And we can get ready for our next question. Some shopping bags can be made from bioplastic. Bioplastic can be made from plant starch or cellulose. You need to describe the differences between the structures, not necessarily the functions, that's not there, between starch and cellulose. We've covered cellulose in our lesson today. You need to go back through your memory and compare it to starch. For 3 marks, give it a go. So starting off, starch is made out of alpha glucose and cellulose is made out of beta glucose. Starch contains 1 to 4 glycosidic bonds, and cellulose also contains 1 to 4 glycosidic bonds. Then we have a difference. Starch also has branched chains because it's a storage molecule for energy, so this helps it make it more compact, remember? So we have starch with branched chains and cellulose with straight chains, or unbranched. And our last point that we could say is starch has no hydrogen bonds between chains and cellulose has hydrogen bonds between them. So score yourself out of 3. Our next 2 are quite quick, 2 markers. First off, we have a definition. Explain what is meant by the term tissue. And after that, for 2 marks, we need to describe the functions of xylem. So, another 4 marks. Give yourself 4 minutes. In our first question, we have a tissue being made of many cells, and that you need to communicate the idea that these cells are going to be working together for the same function, or a common function. Thinking of the role of lignin in xylem next, the lignin is going to be holding these microfibrils together that form that xylem vessel, and you could also say it helps keep the microfibrils parallel. In our next question, we have a 4-mark answer, but there are 8 different things that you could say, so if you want to push yourself, go for it. First off, we say plant cells may contain cellulose. We need to describe the structure of a cellulose microfibril, so we're going to need a bit more detail than what we said in our first question. So let's give it a go. Think of all of the different materials, all of the different bonds, all the different layers that are present. What is going to be going on in a cellulose microfibril? You can do a bit of filtering here. You need to think about what information is useful and is directly related to a cellulose microfibril, and what isn't, and you might be able to save it away for a later question. So, as promised, here are the 8 different things that you could say. Remember, we're focusing mainly on the cellulose microfibril part. So, cellulose contains beta-glucose. We know that beta-glucose is joined together by condensation reactions to form glycosidic bonds. And if we're being extra fancy, you could say they're forming 1 to 4 glycosidic bonds. You could mention the fact that in that cellulose chain, we have inverted alternate glucose molecules, and this means that it stays straight or unbranched. And remember, we're talking about a microfibril, not just one molecule of cellulose. So a microfibril could be made from many cellulose molecules. And lastly, those fibers need to be held together somehow, so we're using hydrogen bonds to hold together those cellulose chains. Out of all of these 8 points, score yourself out of 4. And it brings us to our very, very last question. So, cellulose molecules are held together in bundles called microfibrils. These microfibrils are embedded in a matrix containing calcium pectate. We have seen this before. Look at the words. Look at the structure of it. Where can calcium pectate be found? Is it in the meloplast, chloroplast, the middle lamella, or retonoplast? And hopefully you've ticked middle lamella. So please score yourself out of 12 and upload your booklets to Moodle so I can have a look through your work. As always, if you need some extra help on this, feel free to contact me through email or on Microsoft Teams. And I hope you have a lovely day. See you later.